Humanity has chased immortality for thousands of years. We built myths about gods who never aged. We searched for the fountain of youth. We dreamed of uploading our minds into machines. And today, billions of dollars are being poured into anti-aging research, because death feels like the ultimate problem. But here's the strange thing. We already have immortal cells. Not in the distant future, not in science fiction, right now. Inside laboratories around the world, cells taken from a woman who died in 1951 are still alive. They have divided trillions upon trillions of times. They have traveled farther than any human ever will. They have been to space. They have helped create vaccines. And they may be one of the most important biological discoveries in history. These cells are called HeLa cells, and they reveal a disturbing truth. Immortality is not the opposite of disease. In many ways, immortality is disease, because the thing that kills us is often the thing that keeps us alive. To understand why, we need to ask a deeper question. Why do humans age at all? If some cells can live forever, why can't the rest of us? Every second, millions of your cells die. Skin cells fall away. Blood cells are replaced. Cells in your gut are constantly renewed. Your body is less like a statue and more like a city under perpetual construction. But there is a limit. Most human cells can only divide a certain number of times. This limit was discovered in the 1960s by scientist Leonard Hayflick. Normal human cells divide about 40 to 60 times before they stop. This is called the Hayflick limit. After that, the cells enter a state called senescence. They are alive, but they no longer divide. Over time, senescent cells accumulate in our tissues. They contribute to aging, wrinkles, frailty. Slower healing, declining organs. So what enforces this limit? The answer lies in tiny structures at the ends of our chromosomes, telomeres. Imagine your DNA is a shoelace. Telomeres are the plastic tips at the ends that prevent fraying. Each time a cell divides, its telomeres become slightly shorter. Eventually, they become too short to protect the chromosome. The cell receives a signal, stop dividing. This is one of the body's main defenses against cancer, and this is where the story gets weird. In 1951, a woman named Henrietta Lacks was treated for an aggressive cervical cancer. During treatment, doctors took a small sample of her tumor. Most cells grown in laboratories die after a short time, but Henrietta's cancer cells behaved differently. They divided, and divided, and divided again. They never stopped. Researchers realized they had discovered something extraordinary, a human cell line that appeared immortal. These cells were named HeLa, using the first letters of Henrietta Lacks' first and last names. Today, HeLa cells are everywhere. If you stacked all the HeLa cells ever grown, they would weigh thousands of tons. They have been used in more than 100,000 scientific studies. They helped develop the polio vaccine. They contributed to cancer research, genetics, virology, and space medicine. They are arguably the most famous cells in history. But why are they immortal? Because they are cancer. Cancer is not an invader. It is your own cells breaking the rules. A normal cell follows strict instructions, grow when needed, stop when the tissue is complete, repair damage, die when necessary. Cancer cells ignore those instructions. They acquire mutations that let them divide uncontrollably. One of the most important tricks they learn is how to maintain their telomeres. They activate an enzyme called telomerase. Telomerase rebuilds telomeres after each division. Instead of getting shorter, the telomeres are renewed. The cell escapes the Hayflick limit. It becomes effectively immortal. So the same mechanism that could theoretically grant eternal life to a cell is also one of the hallmarks of cancer. This creates a profound biological dilemma. If our cells could divide forever, we might stop aging. But we would also massively increase the risk of runaway cancer. Nature chose a compromise. Most of our cells are mortal. And because our cells are mortal, we are mortal. At first glance, immortality seems obviously useful. Why wouldn't evolution select for creatures that live forever? The answer is that evolution doesn't care about longevity. It cares about reproduction. If an organism survives long enough to reproduce and raise offspring, evolution has largely succeeded. Living another 1,000 years provides little additional advantage. In fact, immortality can create problems. Old organisms accumulate damage. They compete with younger generations for resources. They may become less adaptable to changing environments. Mortality allows evolution to experiment. Each generation can carry new mutations. New solutions can emerge. Species can adapt. Death is not just a failure of biology. It is one of the engines of evolution. But some organisms seem to have found ways around this. In the oceans, there is a tiny jellyfish called. When stressed or injured, it can revert from its adult form back into an earlier life stage. It effectively rewinds its biological clock. Some scientists call it the immortal jellyfish. Then there are hydra. These small freshwater animals constantly regenerate their tissues and show remarkably little sign of aging. Certain planarian worms can regrow entire bodies from tiny fragments, and some trees can live for thousands of years. Nature clearly possesses many strategies for extending life. So why are humans different? Because large, complex bodies create a dangerous trade-off. The more cells you have, the more opportunities there are for cancer. An elephant has far more cells than a mouse, yet elephants do not get cancer at the enormous rates we might expect. They evolved additional tumor-suppressing mechanisms. Humans evolved our own balance between regeneration and cancer prevention, and that balance is fragile. Modern science is trying to tip the balance. Researchers are exploring telomerase therapies, senescent cell removal, stem cell treatments, gene editing, cellular reprogramming. The goal is not necessarily immortality. It is to extend healthy life, to delay the diseases of aging, to keep people vigorous longer. Some experiments in mice have already produced dramatic results. Scientists have reversed certain signs of aging in tissues. They have improved regeneration. They have extended lifespan, but every step towards cellular immortality raises a terrifying question. What if the treatment also makes cancer easier to create? Because the line between healing and uncontrolled growth is astonishingly thin, and that line may determine the future of our species. When people imagine immortality, they usually picture a healthy person who simply keeps living. But biology rarely works that way. Cells that divide forever often become unstable. Mutations accumulate. Control systems break down. The organism can become less coordinated, not more. In a sense, aging is partly the price we pay for preventing chaos inside our bodies. Your cells are constantly being checked, limited, repaired, and sometimes destroyed. Those limitations protect you. The tragedy is that they also eventually kill you. So the real question is not whether immortality is possible. The real question is whether a complex creature can remain both immortal and stable. And so far, nature has not given us a clear example. But humans are now trying to become the first. What happens if we succeed? And what happens if we succeed too well?